Good afternoon, everyone. It's a pleasure to be here with all of you and with our speakers, and we're looking forward to this session. And without taking too much more time, I want to open with asking everyone here in your companies, how do your foundational technologies and what are you working on, if you could give a description, and how those foundational technologies differentiate, and what is that difference between your technologies, gene editing or gene therapy, and what is the impact of that?
I'll get started. Thanks for inviting us here today. I'm Faraz Ali. I'm the CEO of Tenaya Therapeutics. Tenaya Therapeutics is a clinical stage company focused on heart disease, particularly genetic medicines for heart disease. Of course, heart disease is the leading cause of death in the world. And our fundamental idea is to break big diseases down into smaller genetically defined populations and then use genetic medicines to target them. We do also go after both rare and prevalent forms of heart disease. We're modality agnostic, so actually we have several foundational technologies and approaches. Currently in-house, we have a small molecule. We have 3 clinical stage assets, 1 small molecule, 2 gene therapies. But in our early pipeline, we also have gene silencing, gene editing, and cardiac regeneration. So we have quite a few things going on. Probably one of the things that enables a lot of what we're doing, human iPSC-derived cardiomyocytes for phenotypic screening, AI-enabled drug discovery, viral vectors for delivery of payloads, whether it's a gene therapy or gene editing, specifically to the heart, and even within the heart, specific cells in the heart. So those would be 2 or 3 of the foundational technologies of the company.
Yeah, and I'm happy to follow up after that. So I'm Lucas. I'm one of the co-founders of Mammoth Biosciences and the CSO. So a little bit about Mammoth. We were founded about 7 years ago now, back at the beginning of 2018. And that was really the early days for gene editing starting to mature. And when we founded the company with Jennifer Doudna, the goal really was to tackle some of the big challenges in the field that were keeping us from addressing the vast majority of genetic diseases, right? When you look at CRISPR work so far, even with the progress that's been made, it's still very concentrated in a few different areas. And really the reason for that is delivery, right? How do you actually get these therapies to the right tissue and to the right cell type? And so maybe I'm leading you to think that we're a delivery company, but the way we approached that was actually to think about how can we leverage existing delivery technology and actually innovate on the cargo side, so reframing the question to better leverage those clinically validated delivery technologies. So we went ahead and we, through lots and lots of work that I won't bore you with, made CRISPR systems that are about 1/3 of the size of the original CRISPR systems, namely Cas9. So removing about 70% of the bulk of the protein. And that, of course, enables much more facile AAV delivery with these technologies. And importantly, one thing we've focused on is not just how do we deliver nuclease applications, but also what we term CRISPR plus. So doing much more precise control with things like RT editing, epigenetic editing, and again, doing that all in a single AAV vector. So in short, we've really tried to think about taking controlled kind of bets on technology and trying to leverage the progress that's happened in fields like gene therapy. So to your question around kind of what's the difference, we really see them as kind of 2 parts of a whole to drive these therapies forward.
Great. My name is Raju Prasad. I'm the CFO of CRISPR Therapeutics. And so you've got a good sense of gene therapy and gene editing from both Faraz and Lucas. I think you've heard in a couple panels earlier today that Casgevy is an approved program for sickle cell and beta thalassemia. It's a partnership with us and Vertex as the lead commercial partner. And so at CRISPR, we see ourselves as sort of the leader in gene editing, and we're trying to take the approved Casgevy program and take those learnings and figure out, how do we bring more medicines to patients utilizing the foundational learnings that we have from such a transformative therapy. And so we have 7 programs in the clinic now across various indications where we're leveraging these learnings. And so oncology, autoimmune, type 1 diabetes, and cardiovascular disease, where we're going with both ex vivo cell therapy approaches for an allogeneic CAR T islet cell therapies and in vivo delivery with lipid nanoparticle technology. And again, I think all of us here want to develop therapies for patients. We want to really make transformative impact, and also as a public company, do the best for shareholders. And so at CRISPR, we're looking forward to sort of ushering that next generation of therapies, building upon what's been a monumental approval with Casgevy.
Great. Hi, everyone. I'm Sandy Sita. I'm the Chief Development Officer at Lexeo Therapeutics. Happy to be here, and thanks for the opportunity. I just want to share a bit about Lexeo as a company. We are a clinical stage genetic medicine company where we have 3 clinical stage programs. We have 2 focused in genetic cardiomyopathies and another in Alzheimer's disease and a genetically defined subset of Alzheimer's disease. Our approach is using an AAV vector, and we are more so targeting within the cardiomyopathies the diseases in which we have gene variants that result in loss of function of the proteins, and we are replacing that gene with a full-length copy of the gene to lead to improvement in the protein expression. And in the case of Alzheimer's, instead of replacement, it's actually placement, where we are administering a polymorphism of the ApoE2 allele to lead to changes in clinical outcomes for these Alzheimer's patients. So it's administering a gene in which it's been identified as being a protective gene in the case of development of Alzheimer's.
Okay, great. Thanks for that. So maybe a next question following what you all just talked about is the speed with which gene editing is evolving. Even though it's new, it's cutting edge. It seems like very frequently companies are making distinctions by making new discoveries. Terms like base editing and prime editing get thrown around. Maybe just talk to us about what that really means and how the pace of development influences what all of you at your own companies decide maybe to focus on, and how you consider timetables to be relevant.
I'll go first, or?
Yeah, sure. I'm happy to kick off, since I talked a little bit about these CRISPR plus modalities, and we came up with this term because it is every week, something new comes out. So rather than list off everything, we can just use that phrase. I think a few things to mention that I want to highlight about these technologies. One, I think we're extremely excited about it. This is really what's been promised with gene editing. How do we turn the genome into a Word document, right? Which is the analogy that was used back in 2012, and a lot of these tools actually make that a reality. But I think there's 2 things that really need to be pointed out. One is that each of these technologies is largely built off of CRISPR, and you're fusing other domains onto that, and so it makes this challenge of that I was alluding to earlier, of how do you actually get those therapies to the right tissue and to the right cell, even more challenging, right? So to move these beyond kind of great academic proof of concepts, I think we really do need to think about them in the context of delivery, and that's, of course, how we've approached it at Mammoth, with actually building these off of our ultra-compact scaffolds. But I think the other thing that's important to recognize is that these are new, and all of these have pros and cons. And if you ask the company that's just focused on reverse transcriptase editing or just focused on base editing, they'll tell you all the problems with the other technologies. But I think the way we've approached it is really to think about what are, just from a data-driven perspective, what are the pros and cons? And they're really, you come up with a different choice when you think about it from the disease and the genetics perspective than when you think about it just from a tool perspective. And I think having access to all of those different editing modalities under one roof has really enabled us at Mammoth to be able to do that.
Yeah, no, I agree with what Lucas said. At CRISPR, we're very focused on what's the right approach for the indication, right? There's several different nuances to rare diseases. There's several different nuances to cardiovascular disease, to islet cell therapy differentiation, as well as ex vivo CAR T. For example, we had a first-generation CAR T program that got a very good efficacy response, roughly 20% 6-month CR rate. But internally, we thought, how can we iterate on this? How can we really make this a next-generation product that could potentially usurp the first generation. And so we ran both in parallel. And we made a resource allocation decision last year to go with our second generation program. And with that program, we had a novel screening platform where we looked at hundreds of edits and what edits would actually make the CAR Ts more potent. And when we figured out sort of running the programs in parallel, not only did we have a higher potency in our preclinical studies, the manufacturability was also much better with our second generation programs. And so when you're talking about translating therapies from bench to bedside and getting them into the clinic, it's important to look at sort of the experience that you have with the technology that you've gotten to the place where you've gotten 6 or 7 programs into the clinic, and then iterate on that in ways that you think can get that therapy to have the maximum benefit. So, we're looking at a lot of these next generation things with our CRISPR-X team internally, some of these RT editing programs, long insertions. So to Lucas's point, it's very important to stay on top of this field because it is changing by the day. But at the end of the day as well, there's iterations that you can make to really advance these programs into patients, because that's where you get the real value.
I guess I'll add to that, because we have 3 gene editing programs in our early pipeline. One's been disclosed and the other 2 haven't, all genetic cardiomyopathies. And so these guys are the innovators. They're creating the new tools, the CRISPR Plus, the Cas Plus, and we're the consumers of that. We're not creating new things, but we're using them, and truly the rate of innovation is amazing. And that is both exciting and daunting, because in any given day, we're in touch with several companies and understanding how can we apply that technology to a product. And it's not as simple as just being able to take one type and apply it to all of our genetic cardiomyopathy programs. You really need to match the science to the mutation. And so we have, even in our early pipeline, one that uses simple CRISPR-Cas9, good old-fashioned first generation, but another one that is going to use prime editing, and there may be yet a third that might use base editing. And it's like, how do we deal with that kind of complexity? But that's truly where the science points you to. And then on top of that, the iterations and the improvements are happening. And even in our own hands, even though we're not gene editing experts, but in the process of doing optimization of guides and other things and packaging it into AAV, we come across discoveries that make it better, right? And so part of the challenge is, when do you put your pen down, right? You have a target product profile and you put your pen down, and a few months later you file your IP and you're like, I think we're done. And then you see something else in your labs and like, whoa, that would be better for patients. So I would say it's an exciting time, but it is also daunting from a drug development perspective. And then there's do you go with dual vector, or do you, because prime editing, the current machinery of some prime editors, they're too big to fit into a single AAV. So do you go with a dual vector approach, or do you wait and wait for the next generation that fits into a single AAV that might delay your timelines, but that might be the right thing and a better product market fit if you have something that fits all in one into a single AAV. So it's an exciting time, but it is also challenging to stay on top of it all.
It's an excellent segue, like you're all mentioning that the indication requires the approach to be customized, and we are having the luxury of having technologies that can get there. So, and here we have from the investor side on my right and from the physician side, and I want to ask you this question. How do you pick your rare disease indication? We have several groups, advocacy, patients, sometimes N of 1. How do you really pick the rare indication, and how do you see the transition, the evolution to broader indications, and what are those challenges to be overcome, and how do you get there, to clinic, to approval, and to the next indication that opens other doors?
Maybe I'll start. So, I mean, selection of indications really comes down to 2 factors from our perspective, which really is around unmet medical need, and then where there's a strong biologic rationale. Simply put, it's whether you can make a difference for patients. Where is that need, and where can you improve outcomes for these patients that are affected by these rare diseases? I think you asked the question around challenges as well, and where there's opportunities. Over the last several years, I think really the shift has been overcoming some of the technical challenges, and it's come down to how do we implement this and get this into the clinic, into patients, and getting drugs approved. And I think we really have the opportunity to educate and raise awareness. And I think that's at multiple levels, where we think about bringing disease awareness and awareness around gene therapy to healthcare providers. I think one key aspect is really encouraging early genetic testing, so before patients get to having advanced disease, and where you can intervene earlier and make a difference for these patients. And I think the other aspect is around educating patients as well. With the new technology, it becomes really important to educate patients in terms of the potential benefits, but they also need to be aware of the risks. And I think unless we can educate patients and actually get them recruited into our clinical trials, we really then can only have the opportunity to understand the potential efficacy and better understand the safety profiles of our patients. So I think it comes down to awareness and education to overcome some of the challenges that we face moving into the clinic.
Yeah, I mean, just to add to that, I agree that unmet need is extremely important, right? For Casgevy, it was a perfect alignment of, you had the CRISPR-Cas9 technology combined with Stu Orkin and others, probably some people in this city, that were able to define this HbF phenomenon and BCL11A and how that played a role. And then you had the single point mutation with sickle cell disease that was this perfect candidate that had this alignment of rare disease, unmet need, biological rationale, and the technology that was there to meet it. And so, pushing that through was extremely important, and the early data there really unlocked the value to do that, right? The first couple patients that were treated, Victoria Gray has come out and self-identified, just transformative impact. Last week, Jimmy Olageri, one of the patients, actually Faraz and I were just talking about this, just climbed Mount Kilimanjaro. And it was just a phenomenal feat. And we saw one of our colleagues was there sort of texting us and emailing us the trip and how Jimmy was doing along the way. It was amazing for me and for us to see the transformative impact that our therapy could have for this rare disease patient. And so when we're thinking about translating that into other indications, other rare diseases, it's really with that use case in mind and seeing firsthand what we can do with gene editing technology for rare diseases. And in the introduction, there was the number of rare genetic diseases that are there are massive. So it's not a small anthill that we're trying to build here. It's a very, very big problem that we're all trying to solve at this point.
Yeah, and maybe I'll add, of course, unmet need, 100% agree. Obviously, the foundation for any target that you're going to go after. I think for us as a growing biotech, a lot of it, of course, is also practically how much risk are we willing to take on with that lead program, and how do we sequentially de-risk the technology so we can ultimately start to really address those 7,000 genetic diseases. So our lead program is targeting a gene called APOC3, which some of you may be familiar with from other ASO and siRNA programs. And really, the goal there is there really is not very much biology risk, right? We know that this is a protective mutation that you can make by knocking this out. And for us, as bringing in a new technology into the clinic, it really is trying to focus the risk and not have it be spread out across multiple factors of the program. And then again, to follow up on the question also of kind of what's the big challenge for these programs. I think a lot of it, and this is across the gene editing space, is going to be continuing to build up the data sets and the evidence that there really, really is a need for curative therapies, right? This is a very different way of thinking about medicine than historically our healthcare system's been built for. And I'm obviously a believer, 100% confident that this will be proven out. But when we think about these therapies, we really do have to think about how the patient is going to use them, and not just think about it from like the kind of ideal situation where there's perfect compliance, perfect access, all those factors. I like to use the analogy of, it's like saying that just because we have long-acting insulin, that we've cured diabetes, and it's just not the case. You really need to think about how is the patient going to interact with this treatment, and that's where I think the pull is for these one and done curative therapies, and why it's so important that we do the hard work to actually make these a reality.
Yeah, I agree with everything that's been said so far. So it's unmet need, commercial potential, biology. Now, every single thing, but I'll go a level deeper. Every single thing that we're doing carries major biological risk because this is the first time that people are advancing genetic therapies for these particular mutations. The field of genetic cardiomyopathies is relatively new. It is nascent. So at the time that I joined the company in 2018, there was not a list of mutations that was handed to me and said, like, go. So we actually did, this is a real story, one of the first things we did in the first couple months after I joined was we went through a list of 400 different mutations that were known in the literature. We took heart failure, and then there was hypertrophic cardiomyopathy, dilated cardiomyopathy, arrhythmogenic cardiomyopathy, restricted cardiomyopathy. You'll recognize these terms. And within them, then we looked at the literature, we saw that what mutations were involved, force ranked them based on size of gene, would it even fit in an AAV or fit in a lenti, severity, size, the epidemiology, size of the population, unmet need. So we came up with a pretty rational list, and indeed, sure enough, that list was things that were gene therapy came top of the list because haploinsufficiency, the logic of adding a healthy copy of the gene to produce the missing protein was very sound. Gene editing was still nascent back in 2018. And so we parked gene editing and gene silencing indications where like, this is not haploinsufficiency, we got to get to that later, but we had the list. And then these are the ones where gene transfer should work. And sure enough, the first 2 gene therapy programs we're working on were the top 2 things that came out of that list in 2019. So that was a formalization. And guess what? The first 3 things that we're working on the gene editing side were the top 3 in that list back in 2018, but the technology had matured to a point where we could finally go after those indications with the technology that's available today. So that's for the rare genetic side of things. But when we talk about genetic medicines, we're also trying to go after larger indications. So the big categories, HFrEF, HFpEF, dilated cardiomyopathy, within that, there's a real revolution happening in the field of cardiology. We're kind of taking a page out of the playbook of oncology, where we don't just talk about breast cancer anymore, you talk about the specific mutation and then targeted therapy to that. That's exactly where heart failure is going. And so what we've done is, again, trolled through the literature, understood different mutations. The breakthrough that's happened is you can now use human iPSC-derived cardiomyocytes, use gene editing to introduce a specific mutation of interest, and then you can do phenotypic screening, and you can learn and identify ways to go after those indications. It just introduces the possibility of taking large, and then you can look at genetic validation from the UK Biobank or other sources. And now we're beginning to not only go after the canonical severe rare genetic disorders, but going after the larger ones where you have some human genetic validation from the biobanks, and then we can mimic that in human iPSC-derived cardiomyocytes and see what is the right tool for the job. And it may not always be gene therapy or gene editing. It might be a small molecule. Mm-hmm. It may, in fact, one of our lead programs, it's a small molecule going after a particular target that we think is relevant in HFpEF, but that was identified through our human iPSC work. So when we talk about genetic medicines, we're not only talking about rare diseases, but also a force ranking of the opportunities within larger indications, and to find novel genetic targets with that human validation in a modality agnostic way. So it gives us more than we could possibly do on our own. So prioritizing becomes hard, but we've initially decided to focus on the rare genetic side first, and then park some of the larger indications for later, and maybe for partnerships that we can work on with Big Pharma, because we can't do it all ourselves.
Yeah, if I could just add to that. I mean, I think one of the macro things that all of our companies are going after is a subset of the cardiovascular disease medicine space. And I think that genetic medicines are actually going to become very important in changing that paradigm, to Faraz's point. You know, we have 2 clinical programs with LNP targeted therapies for ANGPTL3 and LPA. So LPA falls in that second category for us, talking about a larger indication. So lipoprotein A is a biomarker that has been shown as to be an independent risk factor of ASCVD. There's a couple outcomes trials from RNA-based therapies are going to read out in the next couple of years to show that knocking it down will confer that, and that'll be an extremely important studies that read out. But we're really excited about this target. It's it's a genetically based biomarker where you sort of get it based on your parents' lineage. We think that you can't be impacted by statins. So this is like the perfect large indication where if we're able to knock down LPA and show a durable benefit, and if that correlates with a reduction in cardiovascular disease risk, that's a huge impact for 11 million patients. And so I think it's very important for all of our companies to note that genetic medicines in the cardiovascular space is going to be a huge paradigm shift over the next 5 years.
So in the last few minutes, we've discussed a lot about gene editing, and I wanted to maybe focus on a topic that I think is of interest to many people, which is cure. So how important is an actual cure by the traditional definition? Meaning you get one and done for forever versus needing to be treated infrequently for whatever the disease is? And how flexible do you think gene editing is to be amenable to potentially needing more than one dose?
Yeah, I mean, it's a good question. From the investor perspective, there's a very high bar. I think the therapies that have been approved and the ones that have sort of generated the most value thus far have been these one and done curative type therapies, even in oncology with Yescarta or Kymriah, Casgevy obviously. But the paradigm I think can shift. I don't think we'll see what the target product profile is for an in vivo therapy for sickle cell disease, and we're obviously working on that. But I think if you're able to safely elevate levels of HbF even to 20%, you're going to make differences in patients. So with LNPs, we see that the ability to redose, and we're going after programs that knock down angiotensinogen, for example. We'll see how the data turn out. I mean, having the ability to have a one and done therapy, I think, is very important for investors right now, and the bar is set there. But as this field progresses, there is the ability, I think, to sort of titrate doses if this is going to be a durable effect. And I'm hoping that the investment community will grow over time in the acceptance of this. But at this point, you have to show a TPP for investors that show this one and done type of transformative benefit at this point, I think.
Yeah, I guess maybe to give a little more nuance there, I think it's hopefully not a cop-out, but it depends, right? It depends on the disease and what the standard of care is. If you're going after something like, Duchenne's muscular dystrophy versus, hypercholesterolemia, it's going to be very different around how curative does it actually need to be. I think, of course, in the ideal situation, you have your cake and eat it too. It's redosable and it's curative, and you don't need to redose. But I think it really does get down to again, what is unmet need, and how is that patient going to interact with the therapy? I think another thing that often comes up that I think is important to think about as well is when we're running these early trials and you have a dose escalation, I think having it be redosable, and Intellia's data I think is quite compelling from an LNP perspective that you can redose these therapies. I think that is a compelling reason for patients that might be in those low dose cohorts to be able to actually get another dose and not be stuck as you might be with an AAV therapy with that initial dose.
I think it depends on the disease of interest and the underlying pathobiology, and whether we think a specific disease is driven by one pathway versus multiple pathways. And I guess I think of that in the context of Alzheimer's disease, for instance, albeit not a rare disease, but there are genetically defined subsets. And ideally one and done would be great, but where they're complex diseases, I think that's where maybe you may have to tackle multiple pathways that are involved in the, excuse me, in the disease pathogenesis. And also I think the target organ of interest is critical, whether it could be a one and done, for example, in targeting cardiomyopathies, for example, where the cardiomyocytes rarely divide. There's very minimal turnover in the cells, where one might expect if you transduce the cells that you can have quite durable effect. So I think one has to factor in, if the one and done, how that plays out within the context of the technology used, the disease of interest, as well as the target organ involved.
I mean, it's in the context of LNPs, redosing is possible. AAVs is where typically there's a challenge. You develop neutralizing antibodies. But I would say even over there, the field is advancing. So I think cure is a big word, and we have to be careful about it. I think it's going to be truly possible in some indications, and I think it'll be an unrealistic term in others, right? If somebody already has some replacement fibrosis in the heart, you might be able to alleviate their symptoms dramatically, but you're not truly going to cure them because you can never replace the fibrosis that's there and the symptoms so it really does depend on the disease what a cure means. But redosing shouldn't be a bad word, and I'm glad to see that there are people who are doing really excellent work to try to enable redosing. In fact, as we speak right now, I think Sarepta is running a study with a particular technology that cleaves the antibodies in a way that allows you to potentially redose. We'll see if it works. It works in the animal models. And I think there's 2 or 3 other approaches I'm aware of. So people are working on redosing for viral therapies, and of course in the context of non-viral, it's less of an issue. So I think we should not care so much about it. I mean, one and done is great. It sounds like a great idea, but what matters is getting to the patient impact. And if it takes you 2 or 3 doses to get to that patient impact and then you're done, then so be it, right? So I'd say what matters most is getting the patient to where they need to be. And if you need to redose to do that, and if it's a year later or 2 years later or 5 years later, it's kind of who cares as long as the patient is doing well. Our goal as a field should be to enable redosing of whatever the technology is being used. And be careful about the word cure. Curative, transformative, but it really will depend on the disease.
I think that the last few minutes, I really want to have in very brief terms, what do you think is in store for the future? We talk about being able to edit, to replace a gene. We talk about identifying an indication, unmet need, delivery methods, viral, non-viral, and beyond the challenges, what is there that we can now do, that we learn from lessons learned, from regulatory, from safety, from understanding what a patient needs, what is important to a patient? What do you think is there in the future?
Yeah, I think for us, it's very important that we get Casgevy with Vertex to as many patients as possible, because I think it's important for investors to understand that, and physicians also understand the benefit that this therapy can provide, and that we can generate a multi-billion-dollar product in this space, in this category. I think what we've spent a lot of time and a lot of resources over the last couple of years now is really, with both our ex vivo allogeneic CAR T platform as well as our in vivo delivery platform, really getting the cost of goods down, because we want to be able to bring these therapies to patients at a price point that is amenable and that the healthcare system will value. With sickle cell disease, it's a great therapy, and we'll get it to several thousands of patients, hopefully. What we're focused on there is let's increase that TPP with targeted conditioning with in vivo HSC, right? Let's really broaden the impact that we can have for patients there. So, what I'm hoping in genetic medicines is that we can bring those therapies to as many patients as possible, and also make it more affordable, so that we're not talking about multi-million-dollar price tags for every single one of our therapies over the next 10 years.
Okay, with that, I think we're just about out of time. Any closing thoughts from any of you?
Yeah, I guess just to follow up on your question, I really think that the floodgates are about to open for gene editing, right? The field's really kind of tiptoed into these initial set of indications and made some safe but smart bets where there's unmet need. And I think yeah, now is really where you start to see, this idea that we have established a tool where you can reprogram it, which is really the promise of gene editing, and you're going to see, many, many more diseases and a much wider swath of indications that are going to start to be addressed.
Okay, great. Thanks, everybody.